So without further ado, I'm going to introduce to you Dr. Zhen Lin, who's a professor of infectious disease in the Department of Animal Science. He received both his BS and MS in microbiology at Fudan University in China and his PhD in animal science from the Ohio State University. He received postdoctoral training in molecular microbiology in the Food Animal Health Research Program at Ohio State University, and he joined the faculty here in 2004. His molecular microbiology and immunology training, together with his expertise in infectious disease, allow him to address a broad range of important microbial organisms significant in animal health, food safety, and public health. Specifically, his research is primarily focused on molecular mechanisms of pathogenesis and antimicrobial resistance in zoonotic bacterial pathogens. Dr. Lin also has long-term experience studying the complex interactions between gut microbiota and the host. His functional microbiome research has led to the discovery of novel targets for enhanced animal and human health. His laboratory has strong One Health emphasis on the interface of livestock, wildlife, environment, and human health. He's published 92 peer-reviewed journal articles and 151 abstracts have been presented in national and international conferences. Dr. Lin has received more than $9.8 million in competitive research grants, primarily from NIH and USDA, for which $5.4 million he's served as PI and $4.4 as a co-PI. He's highly active in professional society and has served leadership roles in various organizations. Recently, he was selected as fellow of the Conference of Research Workers in Animal Diseases. And I am really excited about this talk because this is one of the projects that OHI provided some seed funding for. This is an emerging pathogen, and it is awesome because it's a connection between chickens, raccoons, and humans. And I turn it over to you, Dr. Lin.
Okay, thank you, Dr. Miller, for the nice long introduction. Here I'm sharing my screen. Okay, you can see the screen, right? Okay, hello everyone, and it's my great pleasure to share my research, which is focused on the Albertii with all of you today. First, I would like to thank the UT One Health Initiative seed grant for providing pilot funding for this project. And I also would like to mention that this project is highly collaborative in nature. I have multiple collaborators here from UT and from the other different units. Specific thanks to the Irene, who is a scientist in wildlife services, actually located in Knoxville. I also would like to thank Dr. Li Zhang, who is the faculty at Mississippi State University. He provided me the chicken samples for analysis. Finally, Dr. Shinji Yamasaki, a professor at the Osaka Prefecture University in Japan. He is an epidemiologist and who has been working with different zoonotic diseases. And he provided me a panel of different strands, Albertii strands, isolated from different sources. Finally, I also would like to thank my lab personnel, including Dr. Barbara, Miss Barbara Gillespie, and she's a technician, together with Abby, who is an undergraduate, and Hui Wen Wang, a PhD student. Three of them have spent lots of time, energy, and to isolate the Albertii isolates from different sources. Dr. Ximing Zheng and Dr. Liu Cao provided the bioinformatics training and expertise. So first, I would like to share some background information about E. albertii. This is a gram-negative bacterium that was first isolated from an infant exhibiting watery diarrhea and vomiting in Bangladesh in 1991. The infection caused by E. albertii can cause characteristic attaching and effacing lesions as observed in other AE pathogens, such as enteropathogenic E. coli, ETEC, and enterohemorrhagic E. coli. In clinical lab, and E. albertii is often misidentified as E. coli, and to date, it's still poorly monitored in the clinical lab in many countries. However, recently there are increasing evidence indicating that E. albertii is the emerging and the newest member of the AE family of bacterial pathogens. Most of the outbreak infections actually reported in Japan. The table here showed several major foodborne outbreaks due to the E. albertii infection in Japan. As you can see, dated back to 2003, and several major outbreaks happened in 2015-16. And regarding the possible causative foods, still no definitive kind of information, but there are strong links. For example, for the outbreak in 2011, which may be due to the contamination in the well water, and some other outbreaks is associated with vegetables or the breads and even some meats, the chickens. How about other countries? To date, it's still not many reports. I just mentioned in 1991, that's the first case reported in Bangladesh. Since then, this E. albertii strain was isolated from human clinical fecal samples in Australia and also reported in US, but no peer-reviewed article published yet. It's during the ASA meeting in 2017, we noticed that from the Minnesota Public Health Department. And recently, I found a couple of reports from the Iran and Nigeria. They also reported these organisms associated with urinary infections and gastroenteritis. So how about other source, other hosts? Early reports indicated that from the dead birds, these organisms were isolated. And from the pigs and also some retailed meats, which the kind of at post-harvest level, they reported isolation of the E. albertii from the pork and the chicken, which strongly suggests that food-producing animals are potential animal reservoirs for this organism. And clearly, there are very limited reports and limited studies, with the majority of the reports from Japan. Now the question is, why limited information available for the E. albertii, and why most of the reports are from Japan? I think there are several reasons. The first is that the routine bacteria identification systems cannot identify this bacteria accurately and precisely. Usually, this bacteria has been misidentified as a E. coli. Reliable E. albertii specific PCR primers or methods also needed for the efficient identification of the E. albertii. This method just recently developed by my collaborator, Dr. Yamasaki in Japan, and another group in Japan. Selective medium is also very important. You need a good medium system to differentiate the Albertii from the other organisms. We already have a modified medium system for isolating potential Albertii organisms, but it's still not optimal, and sometimes it's still very time consuming and harder to get the real Albertii from many putative colonies. I will also address that later. And to date, only Japanese research groups, Dr. Yamasaki, I mentioned, my collaborator, and another PI, Dr. Oka, these two groups are working aggressively for this bacteria organism. This statement may be not very accurate, and I can say that my group may be the only one group currently actively working on this organism in the US. We are still collecting more information and to move this project forward. But the take-home message here is that the Albertii associated infections are underestimated based on this information and our findings and the findings from other groups. I was pretty happy to see one review article published in 2019 in the infection immunity. And this review article is focused on the Albertii, this emerging foodborne pathogen. However, the authors listed here, they are from different universities, medical systems in the US, but it seems like they are not kind of actively working on these organisms from any of the aspects. Yeah, perhaps they're due to some challenging issues, like I just mentioned, the efficient PCR detecting method and the optimal medium system. The key information for this review is that epidemiological studies are still severely lacking, and particularly the prevalence and the major animal reservoirs of this pathogen are still not clear. Yeah, and so yeah, this is a great article and indicating the importance of the Albertii. But I should mention this is not the driving force to make me move from other projects and also started this new project in several years ago. Here I would like to give a big credit to Dr. Atsushi Hanenoya, and he is an outstanding junior faculty associate professor working in Dr. Shinji Yamasaki, my collaborator's lab from the Osaka Prefecture University. And he is a great scientist, and he has been working on different pathogens. Particularly, he also developed an efficient Albertii specific PCR method, and also optimized the medium system to isolate the Albertii. And he talked with me several times, expressed a willingness to move this project forward and to do some Albertii work, particularly to compare the strain from US and with that from obtained in Japan. We just, I say, okay, let's go to an easy way, since the evidence suggested that the Albertii likely colonized in the food animals. Why not? Let's just get some fecal samples from chickens and to see if we can isolate the Albertii. So here is the one pilot study we did. We isolate the Albertii from a single chicken farm in the East Tennessee. 20 chicken cloacal swabs were collected, and then the samples were subjected to the PCR and then isolation. Finally, we are pretty lucky. We got the Albertii strains from 6 individual chickens. This strains also confirmed by the PCR and the multi-top analysis. Not surprisingly, and these 6 strains are pretty similar based on our PFG analysis and the later whole genome sequencing analysis. The one thing I would like to mention that all these chicken-derived Albertii strains displayed a multi-drug resistance. And we also demonstrated that the resistant genes kind of carried by plasmid, and this plasmid can be conjugatively transferred to the E. coli very efficiently. We also did genome sequencing and showed that these strains are also closely related to those of human origins. Yeah, I will present this specific information later. And then, switch to the raccoon, right? We know raccoon is important wildlife animals posing a threat to public health because they carry different kind of pathogens. One common is like the rabies, right? But for Albertii, like as listed here, the Dr. Asushi Henenoya, after he joined my lab as a visiting scientist, he is also working on one manuscript submitted to the emerging infectious disease. Later it was published, which reported the identification of the Albertii in many raccoons in Japan. So but even that, I'm still wondering, yeah, how to get the samples from raccoons here. It seems pretty challenging. So I did not kind of move that forward quickly until one day. One day after getting to the office, Dr. Henenoya just rushed to my office and very excited and told me, say, Dr. Lin, and I found a dead raccoon on the road on my way to the office here. And may I get this dead raccoon, and swab and to check? I said, no, that's not good. And that's a bigger compliance issue. But clearly I can see his passion. And I said, okay, what I can do. And then I just did some homework and dropped the email to the Dr. Lisa Muller in the Department of Forestry, Wildlife, and Fisheries. I never met with her, and until now, I haven't met with her in person. But she just responded immediately, and I really appreciated that. Today I would like to take this opportunity to express my appreciation to Dr. Lisa Muller for her support and for my research. She responded to me immediately and make connection to the Irene, the local wildlife service officer, and Irene also very nice and responded immediately and check with me what I want. And because they routinely kind of send person go to primarily East Tennessee, Southern Tennessee, and even Kentucky to collect some fecal samples from raccoons. And then, we have this connection. Now I have a kind of the source, these samples for the work. But before I talk about my pilot screening, I would like to share another information. Asushi Henenoya also talked with me about raccoons, in Japan. And that's one of the reasons why they are interested. He talked a lot of information, and one is that here I want to say that raccoon is a big problem in Japan currently. And I found this book, the Smithsonian Magazine, the title of the article is "The Children's Book That Caused the Japan's Raccoon Problem." This was written by the Laura, and I copied one paragraph here, exactly as what Asushi told me. They said currently there are so many raccoons in Japan. They love to go to the garbage can, and like mentioned here, these raccoons, they steal the goods from vendors and feasting on the crops and even damaging the Asian Japanese temples with their sharp claws and abundant poop. But their invasion started back in 1977 when the kids of Japan were going crazy for a cartoon raccoon named Rascal. And it's very easy to Google. If you like, you can Google. This is a cartoon, this boy, and during the childhood, and he has a friend, a pet. This is a raccoon pet, name is Rascal. This character, this Rascal is very cute and mischievous. So makes this character very popular in Japan. And then the Japanese peoples, and they also even most of raccoons are kind of from North America, transported to Japan, and some raccoons even use some family kind of raise this as a pet. But anyway, it's a big problem right now. The interesting thing is this children's book about the Rascal was written by an American author whose name is Sterling North. Yeah, if you are interested, you can very easily find this information from the Google search. Then let's go back to my raccoon work. In 2019, and I got the 32 samples from the Iran. And actually, these 32 samples already have been stored in the refrigerator for a couple of months, and the fecal samples, most of them already dried. We were not very confident we can find some positive samples, let alone isolating the Albertii. However, PCR analysis showed that we identified 6 positive samples, and then we try to isolate the Albertii from all of these samples. Finally, we got the 3 isolates from 3 individual samples. And here I would like to show you this figure is a PFGE, pulse field gel electrophoresis. The result is to show the relationship of the strands. As I just mentioned, the left side here are the chicken strands we isolated from the Tennessee. And you can clearly see they displayed a similar pattern, which means they basically are the same. Or you can say they have a clonal relationship. But if you look at the 3 raccoon isolates, they displayed a very different kind of PFGE profile, indicating that they are phylogenetically distant, even if they were all isolated from a very small area. Yeah, so this clearly indicated that raccoon serve as a natural reservoir for the Albertii, this organism. And another information I would like to stress on is that the antimicrobial susceptibility assay indicating that all the 3 strands are susceptible for all tested antibiotics. We tested 20 different antibiotics. So they are antibiotic sensitive. So you may still remember that the strain isolated from the chicken they are all resistant. Not all, majority of them displayed multi-drug resistance. So this finding suggested that the raccoons may be the original nature reservoir for the E. albertii. And this organism may be transmitted from the raccoon to the different animals, including food-producing animals, through the ecosystem, for example, the creek, river, or other the channels. So anyway, these are the pilot work data we got before we apply the One Health Initiative seed grant, and we were very happy we got the funding from the One Health Initiative. And for the work, we speculated that chickens and raccoons are the unique and significant players in the dynamic interactions among the enteric E. albertii pathogen, animals, humans, and their shared environment. For this specific project, we have two objectives. One is to examine the prevalence of E. albertii in US poultry production and wild raccoons. Second is to characterize the isolated E. albertii strains using a panel of microbiological and genomic approaches. First, I will talk about the study, the large-scale chicken survey work. We got the pilot study is the kind of samples are from the East Tennessee. However, to move this project more efficiently, we identified another collaborator at Mississippi State, and he has a good connection with different farms in the Mississippi State and Alabama. And on the other hand, we were also curious if this E. albertii could be isolated from the poultry production system in the other states in the US. Then we got the samples from Dr. Li Zhang at Mississippi State. And here just to show the brief procedure, the sample cloacal swab was collected from the individual chickens and then shipped to my lab overnight through the express mail. And then we immediately culture the swab in the TSB media to enrich and to get the culture. Then the next day we just use the culture to run the first round of screening, the PCR, by using a validated primer targeting the CDT gene. If we identify the positive samples like here, number 1 with bright band, and then the corresponding culture we serially diluted the culture and sprayed on the specific modified McConkey agar plates. These plates can differentiate the E. albertii from the other E. coli, as shown here. Typical E. coli displayed around the pink colonies, but the E. albertii just showed those kind of creamy white colonies. This figure is a little bit misleading because this is the ideal situation. For this figure, we just mix the two different organisms straight on the plates. You can see this clearly. During the clinical work and when we tried to isolate the individual E. albertii or putative, this colonies, we just say the putative E. albertii. It's depending on the samples. Most of samples, it's very challenging. We have to pick up more than 10 different colonies, even up to 100. Then run the PCR again to see if there is a E. albertii isolates. And sometimes, yeah, and most of the situations, the results are negative. They are the false positive colonies. That raises a significant challenge for us. And I would like to thank the graduate student, undergraduate student, and the technician in my lab for doing this work. That's a lot of work. Sorry. So for this study, we collected 270 chicken samples. And these chickens are from the 9 different farms. Each farm, we randomly pick up 30 chickens. 6 farms are located in Mississippi and other 3 farms in Alabama. Of 270 samples, fecal samples, the PCR positive samples, there are 43, about 16% rate. And finally, we successfully isolated 12 E. Albertii strains, which is a low rate. And I think that this table can show you the more information about our experience with the samples from different farms. Clearly, for some farms, like the farm 1, farm 2, particularly farm 4, there is a high rate of the positive sample, PCR positive samples. And then we successfully got the isolates. However, in some farms, even if we identified the PCR positive samples, but failed to get the strains. Finally, we got 12 E. Albertii isolates from these 9 farms. And we did an antibiotic resistance profile analysis, and 12 isolates displayed these 12 isolates. Of these 12 isolates, there are 9 isolates displayed a multi-drug resistance phenotype, which is consistent with the finding, the limited strain obtained from Tennessee. And then 9 strains from farm 1 and 4 even displayed high level or intermediate level resistance to the carbapenem antibiotics. These antibiotics has a significance in the clinical usage for human therapy. So that's a big issue. We also did the PFGE as expected, the strains from the same farms, and they displayed the similar PFGE profile. And then we sequenced all the strains and performed the genomic analysis. As expected, the strains from the same farm, they are similar, but from different farms, they are phylogenetically distant. Consistently, the AR genes profile and plasmid replicon types were also similar across the strains in the same farm. To get more information, later we also included 9 diverse human E. Albertii strains, this sequence, for the comparative genomic analysis. Here is the results. Let's ignore this the variance gene pattern. We'll just focus on this phylogenetic relationship. The middle one, the strains in the red rectangle, the TNC1 is the Tennessee chicken E. Albertii strains. I just mentioned we isolated. The bottom 4 strains were from the one farm in I forgot in Alabama or Mississippi, but they were from the same farm. They were all chicken strains. KF1 is a human clinical isolates, and this is the reference strains and the whole genome. The first strain with the genome published. So as you can clearly see here, these chicken strains displayed a very close relationship with this human strain. Then taking a look at the bottom, these strains in the orange rectangle, all these PS strains and the PT strains were the chicken E. Albertii isolated from Mississippi or Alabama. They are almost identical to the one human E. Albertii clinical isolates. And this isolate was isolated in the USA. So this information strongly suggests that chicken is a potential reservoir for the human E. Albertii pathogen. But the challenging part for the epidemiology work is to to establish a solid link, right? It's not feasible to use the chicken strains to challenge the human to see if they can cause a disease. Now, however, we can do another work, like we can use the human clinical strain to challenge a chicken to evaluate their colonization level and persistence time. So later we picked one multi-drug resistant human strains, Albertii strains, and then challenged the chickens, and then picked the fecal samples at different time points post-infection to evaluate the colonization. So as shown here, the human Albertii can colonize and persist in the chicken very well, up to 15 days post-infection. The colonization level is still the average level 10 to 5 CFU per gram of the feces. So this is the data strongly indicated that the chicken is a potential reservoir for the human Albertii pathogen. Okay, let's switch gear to the raccoon. The big question is the raccoon is significant reservoir for Albertii in the US. We primarily focus on the Tennessee and the Kentucky due to the source of the samples. We performed a longitudinal surveillance and the characterization of Albertii in US wild raccoons. This table summarizes our work in the past 3 years. I already mentioned the work, the first batch of the sample, 32 raccoon samples, and we got 3 isolates. And for the second round of the samples, we have a total of 105 raccoon samples. We have a pretty high PCR positive rate and also high isolation rate. I think the reason is I communicated with Irene, and she also modified the kind of the sample collection, particularly storage condition, that improved a lot. And also we, upon available of these samples, and my lab persons just drove to her office to pick up samples. So I think that this is good. Last year, even if we have more samples, but we have a lower positive rate and isolation rate is low, we don't know, maybe related to the lengthy sampling process and the storage. Anyway, finally, we got the 22 Albertii strains from the wild raccoon in the US. And here is just to show you where are these isolated strains from. 9 from the Chattanooga area, and the other from the counties in Kentucky and in the East Tennessee. And first we performed the antimicrobial resistance profile analysis. And here you can see for the raccoons isolated in the US, all 22 isolates are sensitive to all the 20 tested antibiotics. They are sensitive. My Japanese collaborator Shinji and provided me a panel of the Japanese strains, including 20 raccoon isolates. These raccoon isolates, almost all of them are sensitive to the antibiotics. The 7 human isolates, there are 2 displayed multi-drug resistance. We also got 1 swine strain from isolated in Japan. That strain also sensitive. Here is the chicken strains. I would like to mention that again. These 18 isolates isolated from the chickens in Tennessee, Alabama, and Mississippi. Most of them displayed multi-drug resistance. Yeah, we also did the work to isolate the Albertii from the river. We got 2 samples from the Tennessee rivers. Both samples are PCR positive. And finally, we got the 2 strains. However, we did not put more efforts on that because of the challenging. It's the false positive rate is so high, so we decided to just focus on the raccoon isolates to do the additional work. I do not expect you see clearly for this comparative genomic analysis. I just extracted several interesting findings to share with you. One is I just already mentioned. We noticed some strands from the chicken. They were displayed almost the same as those isolated from human. Like this one, these two strands from Mississippi, Alabama, almost identical to the US human isolates. And another two chicken strands has a clonal relationship with these human isolates, originally isolated from in Poland. Yeah, so that's one finding. Another is we want to know if we can see some clues, the connection between the isolates isolated from raccoon and human. And we did find that there are highly similar strands in those from Japan, because I think Japan, they have a good comprehensive collection. They not only collected the isolates from raccoon in that area, but also in the meantime, they have a wide collection of human isolates. As shown here, these two, one from human, one from raccoon, and the right panel, one from raccoon, one from human. These two, they are almost identical. They're strongly indicating that raccoon could be a source for the human infection. And then another question is, can we find a similar E. coli isolated from the raccoons in Japan and isolated in the US? Yeah, the answer is yes. We did find the raccoon isolates in the US are very similar to those isolated in Japan. And we also found that the Japan swine strain also has a very close relationship with a couple of Japan raccoon isolates. And then the big question is that, do we have any information about the Albertii transmission between the wildlife and the livestock in the US? Simply saying no at this stage. For our project, because of the chicken isolate from the Alabama southern area primarily, and the raccoons are limited to the Tennessee, so it's hard. We did not find any similar, but I speculate that if we have a well-controlled study, and we should be able to find this kind of connection. Now that we speculate, I just mentioned for the strains isolated from the raccoon, they all display the very susceptible phenotype to the different antibiotics. So this is interesting. But the strains isolated from the chickens, most of them multi-drug resistant. So based on this, we speculated the raccoon is a native, original nature animal host for the E. albertii. And this organism may transmit, it's a bidirectional, I believe, but maybe originally from the raccoon to the other animals, particularly if they are close to the animal production system, I think that may work. So to test that, we did another animal studies. We use 3 diverse raccoon E. albertii isolates to challenge the chicken. We did a chicken challenge work, and we observed that the US raccoon E. albertii strain can colonize the chicken intestine. Not very well, but you can see still decent colonization levels by 7 days post-infection at the 10 to 5, 10 to 6 CFU per gram of the feces. However, consistent for all 3 strains by 14 days post-infection, yeah, we barely isolate the Albertii and from these chickens. So this indicated that the raccoon Albertii can colonize in the chicken intestine, but may not persist as long as the human isolates. If you think about this dynamic, how the chicken production systems, if the isolates colonized in the chicken effectively and then disseminate to other chickens and then pass along, I fear this eventually the strains may acquire the persistent colonization in the chicken intestine. So here is this is a general model. We are working on that. We fear we need a One Health approach to control the Albertii. And however, to address several important issues and to get the more definitive information about the evolution and ecology of these important strains, pathogens, I think we need a well-controlled studies. And for example, like to get identify the chicken farms in Tennessee, and in the meantime, we have the samples, the raccoon samples from Tennessee and with the adjacent rivers, and also with the collaboration from the public health department to get the human isolates. Then we may get have a much better clue about this dynamic interaction. So, of course, as I mentioned at the beginning, that definitely needs a teamwork. Yeah. So, I think that's basically all I have today for you. But before going to answer some questions, I would like to share a video with you and a small video clip. Yeah, I know all of you in the past 2 years and have been going through the COVID pandemic. So I think we need to put some pandemic spice here at the end. I'm just wondering what we can learn from the raccoons. And I got this video clip, and you can see. Well, yeah, clearly, yeah, hand washing is very important, yeah, together with face mask. Yeah, that's a important prevention measures for us to deal with and this COVID pandemic. So, okay, and finally, I will be very happy to take any questions from all of you, and that's all. Yeah.
Thank you, Dr. Lin. That was wonderful. Everyone, you're not able to unmute yourselves if you'd like to answer or if you'd like to ask a question, or you can also use the chat feature.
Okay.
I actually have a question, Dr. Lin.
Okay, what is that?
Yeah, so first of all, a very, very nice study. Thank you. It's very, very interesting and nice and good work. And my question is, E. Albertii in humans, does it cause a typical EHEC or EPEC lesion, or it just has only does it also have like intimin, those genes for tight attachment, like intimin in the tear?
Right. They have a panel of, but simply saying yes, the infections based on the limited reports from the human, primarily from the Japan, they created the typical AE lesions and bloody diarrhea. Yeah. But one thing is I want to mention the molecular pathogenesis work and which are the primary virulence factors for this pathogen still unknown. It's not clear.
Yeah. When you infect chickens with a raccoon and the woman isolate, did they cause any lesion on the intestine?
Oh no. Chicken is totally normal. Yeah.
Okay. Okay.
Yes.
I have a couple of questions. So
Sure, go.
first one is the species identification. So you mentioned E. albertii tend to be misidentified as the E. coli. So for the current state, what kind of method you used for species identification? That's first question. Yeah.
Okay, let me quickly address this. And as I mentioned, in the general clinical lab, I think maybe this statement, like in Japan, they may not kind of traditionally that method cannot differentiate the E. albertii from the E. coli. And however, recently there are several PCR method, particularly the one method developed by my collaborator that targeting the CDT gene, which is very specific and reliable to differentiate this from the other kind of similar organisms. So that is very helpful for the original initial screening. However, we had one question is that it was indicated that this organism may be misidentified as E. coli. So how about the current library, a big library of the E. coli strains and from the animals, and can we find some E. albertii or misidentify? So to address that issue, several years ago, I requested the strains from my other collaborators, and which I think about 200 E. coli isolated from pets, those provided by Iowa State Vet School. And I also got about more than 500 E. coli strains isolated from chicken, which provided by the University of Georgia. And I got more than 300 E. coli isolated from the beef cattle from the Nebraska. And we did the PCR screening. And we only find a couple of misidentified the E. coli, but not E. albertii. However, we did not find any kind of E. albertii from those E. coli strains. So I fear that I didn't go kind of deeper to check their protocol for the isolation of the E. coli, but I kind of I'm confident that the current kind of protocol used in these three different institution systems, at least the E. coli they identified isolated are true E. coli. Yeah.
So well, you kind of answered part of my second question. So the identification of microbes nowadays seems like the multi-locus sequence typing is the kind of the commonly used method. So do you have the sequence data equivalent to E. coli for your albertii, so you can make some comparison to see if, say, their misidentification use multi-locus sequence typing, to what extent that can be? So maybe something interesting to look at.
Okay, yeah, I think that, but the point is multi-locus, that's a typing method, right?
Yeah.
It's not a typing method. I think currently, yeah, gold standard, we use the PFGE, and for these enteric pathogens like Campylobacter, Salmonella, E. coli, but now with the availability of this kind of genome sequencing, and we can have a much more information, and and we can easily differentiate the kind of this Albertii strains from the other E. coli strain. That sequence information very straightforward. But in the clinical lab, I think maybe the feasible way, one is the PCR using the specific primer. Second is after getting the potential isolates, you can use that MARDI-TOF that commonly used in different vet school right now, and you can use that to do another level of the validation if you don't want to do the genome sequencing. Yeah.
Thank you. Well, I have another question. If no,
sure,
no
sure.
So based on your sequence data between Japanese sample and the samples from America, is it fair to infer the Albertii was transmitted from US to Japan?
I cannot say that, but it's kind of that's one thing is I think this one here, and we also should take into consideration for the import-export this channel. And at least one thing I'm confident, originally, because of that cartoon, the many raccoons in Japan, they were from US. Yeah.
Okay.
So any other questions? So anyway, I would like to thank this One Health initiative again for supporting this project, and that's really critical for us to move the work forward. But as I mentioned, there are still lots of limitations over there, and I hope in the future we can have some good opportunities and to submit a team proposal and to address some key questions. Yeah.
Thank you so much for a wonderful talk. And we too are looking forward to seeing you take this to the next level.
Okay, thank you.
And thanks everybody for attending. Hopefully you marked your calendars for future events. Thanks.
Thank you.
Thank you so much.